Welcome back, Year 12s. Today we're looking at the second line defense as well. So we have cells, we have proteins, we have other responses that we're going to look at, including MHC1 and 2 markers, which are found on the cell membrane of all our cells. MHC1, of course, showing that we are self, and MHC2 found only on our special APC cells. Many of our immune cells are also phagocytes too, which means they engulf different things. Those include neutrophils, macrophages, and dendritic cells. We're continuing on with our current theme of immunity in the second line defense today. All right, so our next cell we're going to look at is the natural killer cell, or the NK cell. So they're large, all right? They work on destroying cells that have been infected by viruses, or cells that are damaged or cancerous, okay? So if we're looking here, all of these cells here could be cancerous or soon to be cancerous, all right? Or they could be infected by viruses. Essentially what they do is they release what we call death ligands, all right, which will then cause the cell to go through apoptosis. So these cells, they use the MSG1 markers and killer activation receptors, and they will check each and every cell to see if they want to make it go through apoptosis. What happens with the NK cells is that they will check for MHC1 markers as well as this killer inhibitory receptor. All right, if that connects, all right, that is a self cell. If that doesn't connect, that cell is going to be destroyed. All right, our next cell is the eosinophil. Okay, interesting name, but what are they? They're large granulated cells containing various toxic chemical mediators such as DNases, RNases, and proteases. Again, they are all, as we should know, enzymes. Okay, anything ending with A's is an enzyme. So breaking down DNA, breaking down RNA, breaking down proteins. Of course, what's that inclusive of? Multicellular organisms. All right, so these cells are not really efficient at getting rid of most pathogens, but when it comes to a larger multicellular pathogen, it could be a parasite, it could be a protist, but generally that's going to be your parasitic worms. All right, so stuff like ringworm, tapeworm, all of those things. So why are they necessary, eosinophils? Because essentially these multicellular pathogens, all right, like a tapeworm, it's way too large to be phagocytosed. So it's way too large to be engulfed by your white blood cells, all right, by the different phagocytes. So they degranulate on contact and release their chemical mediators, and that starts to break down the multicellular organism, all right. All right. Lastly, we have a few different types of different molecules you'll find in the immune system. So our first one we're looking at is what we call an interferon. All right. And that's a signaling molecule. So it's a type of cytokine. So it's a type of cytokine. That's your interferon. And it's literally, what it does is in its name. Interferon, it interferes. So it interacts with MHC1 markers on neighboring cells. If a cell has been infected, okay, and if that cell's infected by some sort of virus, it's gonna, once that bursts, which it will do, all right, these viruses will spread out and try to get into neighboring cells. All right, that's what they do. All right, and in that case, these neighboring cells, they have interferons which come in, as you can see, and they interfere with the protein synthesis of that cell. So interfering with the protein synthesis makes them not create as many viruses. So the spread isn't as bad through your cells. Okay, and that's really important. So they interfere with the protein creation of neighboring cells when there is a virally infected cell. The other molecule we look at is complement proteins. These ones are a protein. So they float around in the bloodstream, all right, and they're made by the liver. Another reason the liver is super important in the body. They act as a chemoattractant, so they chemically attract phagocytes to the area, so it can get rid of stuff like bacteria, as you see here. They opsonize the bacteria by flagging them for death, so they jump on them, and essentially they also signal phagocytes. So opsonization chemoattractant is similar, all right. And they can also make an MAC complex, which essentially breaks down the plasma membrane of this bacterium, and the cellular contents leaks out. And once that leaks out, all right, bacteria dies. All right, no good. So complement proteins are really important too. They are a really underappreciated and under-remembered protein in the immune system. Okay, so interferons and complement proteins very important too. Thanks for watching.